So with that, I'll finish my introductory remarks and I'll start the introduction for Joshua, a very important person whom I met in 2018 here in Boston. And our meeting is a great example of the benefits of in-person meetings. It was at the Festival of Genomics conference organized by Oxford Global. And I heard there the presentation of this really interesting instrument that Josh was talking about doing single-cell sample prep. They talked about genomics, but that very same year, Harrison Specht, a graduate student in my lab, had made a lot of progress on developing our minimum sample preparation method, and they knew how much there is to gain from having good sample preparation method, and they knew that there was more space for improvement. So I was really excited to hear about the capabilities of what Joshua was talking about, and a little bit skeptical because he was wearing the hat of a salesman, sort of. He was talking from a company perspective. So I approached him with some skepticism later on in the sponsor booth. And Joshua very quickly dispersed my skepticism because he sounded like an engineer. He wasn't interested in just selling me something. He wanted to understand what my problem is. And then he started talking about the technical specifications, and he talked about the very chips that ultimately have now turned into the ProteoChip that is used with CellenONE for proteomics and for other applications. And even from that very first meeting, I thought it would be awesome to work with this engineer. And that first impression has very much solidified over the years. So later on, we kept inviting always Joshua and his team to our conferences, which has enabled other colleagues to also begin using the CellenONE instrument for various applications and sample preparations. And today, we are very fortunate to have Joshua come and to give us an introduction to that very powerful platform for manipulating single cells in very small volumes, but very high throughput. Welcome, Joshua.
Well, thank you so much, Nikolai. Well, thank you. Thank you so much for that wonderful introduction. I was actually just up here. Yes, thinking about that day in 2018 when we met, and I had never heard or thought of single-cell proteomics from a an untargeted manner. We're familiar with the fax machines and other ways to look at some of these things, but you really ignited a fire in me and in all of CellenONE, really, to just understand what is needed to make this a preparation that people can use to better understand biology and to guide precision medicine, hopefully in the end, which I think is one of our big goals. Right here, just down below that. So today I'm going to introduce you to the CellenONE, our automated single cell isolation and ultra low volume dispensing instrument, and how it can be used to achieve easier, faster, and more reproducible proteomics data generation from single cells. All right, our company is CellenONE, and we provide tools that enable gentle and accurate dispensing of single cells and reagents for multi-omic studies as well as cell line development. We're a spin-out company of CellenONE, whose core competency is ultra-low volume precision dispensing, and we are now part of the CellInc group, world leader in bioprinting and other precision medicine enabling technologies. The CellenONE can deliver high-quality single cells for many downstream applications. In addition, it can dispense reagents and control the temperature and humidity of the working area to help automate many workflows. It accomplishes this by first automating image-based single-cell isolation in a very gentle.
Yeah.
Oh. I like this kind of engagement of our audience. We'll have time for questions at the end, and I look forward to yours. So yes, we do single cells really well. Gently viable cells onto almost any surface, of which you're going to see a number of those different targets today. And secondly, we dispense these very low volumes of reagents in an automated manner, and we can complete all the steps for preparing single cells and multiomics workflows, such as single cell proteomics. The core dispensing technology was perfected at CellenONE over 2 decades for the production of FDA-approved diagnostic devices and is driven by a piezoacoustic generated wave in a structured glass capillary, which allows high-frequency drop-on-demand generation of picoliter volumes from a variety of reagent classes with low variability. Our single-cell isolation is enabled by this reproducibility of dispense and onboard imaging analysis of how cells travel through the capillary. To adapt this amazing technology for single-cell isolation, we developed an image-based single-cell isolation mechanism that works like this. We segment our nozzle into 2 zones. The ejection zone, which is determined empirically as the volume containing cells that will be ejected in the next droplet, and a sedimentation zone, just above this to avoid any cells sedimenting into the ejection region prior to isolation. Here you can see the CellenONE operating in real time. When a single cell meeting your isolation criteria is detected in the ejection zone and nothing in the sedimentation zone, the robot moves to spot the cells in the plate. All empty droplets and droplets including multiple cells, like the one shown here, dispense into a recovery vial, enabling high recovery of rare cells. With an optimized sample, the CellenONE can fill a 96-well plate with single cells in less than 3 minutes. The CellenONE can handle and isolate a wide variety of morphologies, from bacteria when using our 4-channel fluorescence-based isolation to even very large cells like cardiomyocytes. By combining high accuracy cell isolation and picoliter to nanoliter volume dispensing, we are enabling a wide variety of automated and miniaturized single cell workflows. In the next few slides, I'm going to provide a few examples of some of the applications that are currently being enabled by the CellenONE. One of the main advantages of the CellenONE for cloning workflows is perhaps best demonstrated in cell line development, where the health and viability of the dispensed cells is hugely important and can be measured by the subsequent clonal outgrowth from these single cells. Here we show that our single cell isolation and outgrowth is not negatively affected by well sizes down to even 1536 well plates. Moreover, the ability of the CellenONE to process multiple cell lines without user intervention through our automation and API allows integration into automated work cells. For antibody discovery applications, there are very cool workflows for that too. And if you're interested, you should come talk to us. The other major advantage of the CellenONE is found in the omics applications, where being able to dispense into any format and subsequently liquid handling in very small volumes can help with single cell studies in genomics, transcriptomics, and proteomics. It's not difficult to imagine that the CellenONE can also enable things like metabolomics, lipidomics, and other multiomics as well. This is quite significant because it allows different teams within academia or medical research facilities or biopharma companies to get more value from their capital equipment investment. One example in the field of cancer genomics, the BC Cancer Research Center developed a whole genome sequencing protocol named DLP Plus using the CellenONE. This protocol has enabled single cell profiling of multiple tumor types at low cost and has been replicated at multiple cancer research centers worldwide. For single cell RNA-seq studies, we have developed a kit leveraging our cell-on-chip platform, which allows for the preparation of over 1,500 single cell libraries to be generated in nanoliter volumes for many single pot protocols. In this example, we analyze the differential expression of single cells from the same cell type, cultured both in standard 2D cell culture plates and also as spheroids with our 3' RNA-seq kit. Even with only a few hundred cells in total, we were able to cluster multiple cell populations and identify the presence of highly expressed liver metabolism pathways in the spheroid culture that were almost non-existent in their 2D counterparts. This is one of the reasons that 3D cell culture has become so interesting recently. Now, to focus on what brings us here today, single cell proteomics. Single cell proteomics is certainly more challenging than traditional batch proteomics, as anyone who's tried it knows. And I've heard more than once that groups have difficult times getting good results with the plate-based approaches developed in the original scope protocols, due in large part to the small reaction volumes and poor performance of FACS isolation to these small volumes. Fortunately, our technology has been adapted to automate nearly all of the single cell isolation and subsequent proteomics workflows, where we focus on gently dispensing high-quality single cells and miniaturizing the reaction volumes for both unlabeled and multiplex workflows. Here again, the quality and viability of the single cells is critical in order to make sure that all that effort you put into the sample prep and LCMS analysis and data processing is based on healthy and viable single cells. Earlier this year, 3 fantastic preprints were produced by 3 great groups, where each used a different sample preparation target substrate to generate LCMS-ready samples. Here, I'll speak briefly about our ProteoChip, developed in collaboration with Carl Meckler's group at IMP, and the nested Nanopots platform from Ying Zhu at PNNL. And in the next talk, Andrew will give you a deep dive on the developments of the NPOP platform. For multiplex experiments based on the SCOPE protocol using TMT labeling, there are multiple steps for the sample preparation, requiring a lot of manual handling of the samples, in and out of several instruments through the process, slices of the cell, digestion of the protein to small fragments, TMT labeling, quenching, and then also pooling. It has been our goal to miniaturize the sample volume and reduce the number of manual steps required to produce high quality and reproducible mass spec data. That's why we worked hand in hand with our collaborators at IMP in Vienna to develop a substrate that could help in overcoming some of these difficulties. The microwell chip we developed is called the ProteoChip, and it is the size of a microscope slide. It's composed of 12 arrays of 16 wells each, so you can work with up to 16 TMT labels. On the deck of the CellenONE, you can work with up to 3 chips per run, enabling more than 500 single cells to be processed in a single run. Here's how it looks in the instrument. You'll also notice a 384 well plate on the right. This is where you can aspirate your samples with dead volumes less than 5 microliters. There's also a position for Eppendorf tubes on the left, and the CellenONE is really compatible with most laboratory labware for sample uptake. There's also a second camera you can see mounted here to the left of the robotic axis that allows for accurate drop dispensing to microfabricated targets with tolerances less than 20 microns. And also allows for quality assurance and other inspection. When working with the ProteoChip, each well begins filled with oil. The oil layer protects the very low volume samples from evaporation when performing sample preparation steps that require heating. All reagent addition steps and incubation steps are carried out on the CellenONE without need to manually handle the ProteoChips until pooling, which is carried out via centrifugation. Here you can see the CellenONE isolating single cells to the ProteoChip, one well at a time. Next, we'll see the TMT labeling, where we're dispensing nanoliters of TMT label to 1 well of each array. This is done in series where we wash out samples in between and fill the next one. Once the ProteoChip is finished processing, it couples with the ProteoChip funnel. You spin it down. This is the pooling process, and then this can be interfaced directly with your auto sampler for easy injection into your LCMS. This workflow is successful in identifying over 1,000 proteins per cell, even with no carrier channel. And for more information on this protocol, please see the BioArchive paper, visit our website, or come talk to us after during the booth sessions later today. Now, I'd like to give a brief intro into the Nanopots technology for those not familiar, and then some of the developments coming out of this wonderful collaboration with Ying Zhu and his team. The original Nanopots preparation was done using a FACS for cell isolation, a custom liquid handler for reagent addition, and another custom robot for auto sampling that enabled reaction volumes of 200 nanoliters and a greatly reduced adsorptive losses of peptides to reaction vessel surfaces. And while this was a big advancement relative to plate-based approaches in a number of ways, this was still only accessible to a very small group of scientists who had these custom robots. There were losses expected by the pooling of labeled samples, and the throughput was limited by the accuracy of the FACS to deliver cells to the Nanopots wells. So during the beginning of our work, we evaluated the capability of the CellenONE to load a variety of well sizes to understand how densely we could pack the reactions. We validated near 100% single cell loading of a range of well sizes down to 300 microns, allowing a complete reconfiguration of the nanopots. Here you can see the first generation of the nested nanopots, or N2 slides, enabling up to 243 cells to be processed at a single time. This work was driven by Jongmin Wu and with the enthusiastic support of Lily Pasatolek. Single cells were sorted onto a dry N2 slide, followed by addition of all the reagents in volumes of tens of nanoliters. The custom auto sampler was replaced by a simple pipetting method to pool cell samples, making the entire process possible without any instruments other than the CellenONE. Now I'll quickly share some data from this upcoming publication. Here you see relatively good proteome coverage. Even when accepting only a 70% valid value, we produce a clear differentiation of the 3 cell types produced. We also analyzed the published data set for 2 of our 3 cell types and did a Pearson correlation, resulting in a value above 0.95 for both the single cell protein data sets and below 0.7 for the RNA-seq data. We also looked at the coefficient of variation and see again that proteins across the single cell data sets are very stable for samples from cell culture under the same conditions. We took this a step further, looking at potential correlations between RNA-seq and proteomics data, and see that the correlation is really poor between these, generally below 0.3, which suggests that we cannot conclude protein abundance based on RNA-seq data. We all know this at this point, but it's nice that we are still confirming this. Of course, we can start, until we can start getting RNA and protein from the same single cells, we will not be able to understand how these 2 biomolecules are truly correlated. And this is why I'm so excited to share an open call for someone with a background in RNA sequencing development to join our team to help us figure this out in Washington State. And with that, I'd like to thank you all for your time and now welcome any questions.
Wonderful. Thank you, Joshua. And I see that we have quite a few questions. Hopefully we'll have time for all of them, but if we don't get to answer all of them, I'll just want to remind you that you can keep asking questions at 3 o'clock Eastern time during the sponsor booth sessions. So let me get to the questions. So the first question is, tell us about the maximum number of proteins detected from single cells isolated.
It's a good question. It kind of depends on the prep and the mass spec and the LC and the cells. I don't know what the record is if we were to be in the Guinness Book, but and I would have to say that I am not the right person to answer that question in technical specificity, but it's over 1,500. I would.
I agree with that assessment. So it's certainly well over 1,000, and the aim is not necessarily to maximize that per single cell. You're going to hear a lot more about this question later throughout this meeting. The second question is how easy it is for a proteomics facility with no FACS experience to welcome a CellenONE for single cell proteomics analysis.
That's a great question. So I've done work with RNA sequencing groups and biologists and genome sequencing groups, and now a number of mass spec groups. And I think that, I don't know what it is, but the people operating mass specs understand small capillaries. They understand low flow. They kind of get this to start with. And we provide 2 days of training and good support, and you can be up and running in a week generating single cell proteomics data.
So I can also chime in and speak from the side of a user, and I can say that in my lab, we've had many more challenges using fact sorters than using CellenONE. And one of the really great things about CellenONE, even outside of the preparation methods that Joshua described, is its single cell isolation capability. The single weak link in our minimal sample prep method that Harrison has developed is cell isolation. It's the fact sorting. It's not anything else. CellenONE just does a very good job with the cell isolation, and that simple function is very easy to master. Obviously, as anything else, there are different gradations of mastering the capabilities of the instrument. It might take you a while to get to the virtuosity of Josh, but getting started to just isolate single cells is really easy in our experience. Another question, how does an LC-MS chromatogram of a single cell analysis look like? We have exactly an LC-MS chromatogram in our preprint. So look at Leduc et al. Please tell us about plants where single cells have cell walls and it's hard to extract.
So we're currently doing a little bit of work on this for a DOE project, and plants with cell walls. I've seen some very elongated kind of root cells pass through the CellenONE nozzle relatively easily. But if you're interested in what's inside of the membrane, inside of the cell wall, and you can dissolve the cell wall, then the protoplasts are relatively easy to handle. There's a lot of variability in size. So we actually have a new instrument coming out named the CellenONE for handling very large particles, spheroids, 500 microns. And so certainly I think we have some solutions and are working to develop those workflows to make that easier to do as well.
Yeah, and I'll just quickly say that there are a lot of sample preparation methods that they know of that should work very well coming up, that should work very well with plant cells and any other cell that is tough to lyse. There are some sample prep methods that use heavy duty SDS and acidification that should take care of any cell wall. So I'll finish with two more questions before I welcome Andrew. The most frequent question on the chat is, how much does it cost?
So the cost is less than a new mass spec and also less than a new fax machine. So the price is somewhere between 250 and 350,000, depending on what if it's fully loaded or not.
Good answers. I like the preface. And there's a question I see, John. So let me just finish with that, and then I'll give you a chance, John. Sorry?
It's a follow-up what you just said.
Okay.
John Wilson, I'm at Proteo. When will you reach an economy of scale when that can be dropped down, assuming that single cell work bears fruit for example, as you mentioned, clinical applications?
Yeah, so I think that we do want to make this technology available to more and more groups. Right now, the instrument is an industrial piece of automation capable of running, 24 hours a day. So over time, as we really kind of narrow down what the workflow is, we'll certainly look to make it more affordable so that people can do this at a broader scale with less capital costs. But we're also offering kind of reagent rentals, and we really like to work with groups who have great ideas and really enable them to take that leap one way or another. So.
And final question, when is the ProteoChip going to be commercially available?
Not soon enough, but before the end of the year. So.
All right.
Thank you so much.